manufacturing. And because we are a little bit pressed for time, I will go only briefly over their activities and what is going on. So in both situations, we have basically public-private partnerships with the support of the European Union, and that's the European Institute of Technology, and also with private companies, universities, research institutes, and others who are forming this huge network that is collaborating in creating innovation. And it is doing so. So each network is doing so by giving support to startups, by giving support to innovation projects, but also bringing different members together to create new initiatives. One of the things that we do is that we are the hub in Croatia, and the idea is basically to connect innovation community in Croatia with the rest of the network. So to promote the network activities here and vice versa, basically to promote our innovators, researchers, and entrepreneurs to the network. There are quite a lot actually of opportunities for funding. So if this is something of interest to you, I would really advise that you contact me and we can go into them later on, because now there is unfortunately, there is no time to go over them in detail, but there is definitely an opportunity to fund proof of concept projects. There is also opportunity to fund larger innovation projects. So as a part of the European consortium and with EIT Health partners. And also EIT Health has this very developed array of programs in supporting entrepreneurship, from very, very early stage to the then middle and validation stage to really market entry and global scaling. So really, wherever you are in this process, you will have an opportunity to engage. The EIT Health is also running a lot of programs that are aimed at students, and one of the most famous one is the Innovation Day, basically Europe-wide competition. So that where people from, I think, 34 locations in all over Europe participated. The winners are Winners Weeks, and some of you maybe joined us because you are part of the Winners Week. Is happening right now. Finale will be early next week. And we are also happy to organize here locally for our winners a local demo day, which will happen on December 10th. So basically, there are opportunities for all involved, whether you're a student, whether you're a researcher, or whether you're an entrepreneur. And then I will also mention the second kick that is supporting this event. And very happy to be organizing this as a collaboration between two kicks. And this one is really dedicated to manufacturing and to manufacturing innovation. And you will see that here as well, it is very, very important that we have this interplay between between EIT and all the partners who are part of the EIT manufacturing network, who really are participating from different industries. So it is not industry specific, but basically really open to everybody who is interested in innovation in the manufacturing process. There are some, there are some unifying themes that really bring the community together, and this is competitive manufacturing skills, manufacturing innovation, resilient manufacturing, something that we are finding out is very important right now in the pandemic situation. Of course, environmentally sustainable manufacturing, and of course, digitalization. We are part of the Manufacturing East network, but really the idea is to connect the whole European community together, and then and to be able to participate in joint projects and to drive manufacturing innovation together. EIT manufacturing is also very strong in supporting startups. There was just a competition that is still happening as well. The finale will be actually in 2 weeks, for all the different startups, teams, or actually established companies who are looking for either developing or looking for manufacturing innovation. There is also opportunity to connect with other partners and to actually apply. Sorry, I think I lost. Oh, I lost screen share as well. So this is new technical difficulties, but I was getting to the end of my presentation. And there was this chance to connect with with different partners, and also for together then to apply for projects. And actually for this final stage, we have a matchmaking event on the 14th. So if you're interested in that, we will send you a link later on. And this is basically all about my with respect to my introductory presentation. I will, unfortunately, you didn't see my email, but we can put it in the chat so that if you think, if you find any of this interesting and you are interested in participating, whether it's the entrepreneurship program as a student or as or already as a startup founder, then feel free to contact me, and we can, we can help you out with information how to apply and how to best engage. And now really we will move to the rest of our event, to the rest of our session, and very happy, that we have with us really an array of amazing speakers, which I hope they are here. I know they are here signed up, who come both from the pharma industry here in Croatia, but Croatian companies with global presence and global connections, and also are the members of the academic community here. So the first speaker is Aleksandar Danilovski, and Aleksandar is coming from Xellia Pharmaceuticals, which is a global company with a headquarters in Denmark, but research and development center in Zagreb. And Aleksandar is the chief scientific officer with, I would say, over 20 years of experience in pharma industry, and he's here to give us an opening session. So please, Aleksandar, if you can connect and share your screen with us.
Thanks, Vlatka, and hello all. Let's do this. Okay, share screen. Can you see the slide? Great.
Yes, I will just, sorry, Aleksandar, I will just intervene very briefly because so that all the participants know. Unfortunately, Aleksandar has to leave us at 4 p.m. because he has another meeting, which really came up very suddenly today. So Murphy's Law in action. So please, if you can ask questions in the chat, I will see them, and then I will be able to ask a couple of questions after his presentation. So we will not wait for the Q&A session. So feel free to ask questions in the chat. Thank you. Aleksandar, sorry, please go ahead.
Yeah, thanks, Vlatka. And it's for the menu, I saw a couple of names that I recognize. Aleksandar is too official, it's Sasha. So hello all and good afternoon. I'm really happy to be on yet another winter School of Pharmacy, which is turning into a traditional event, which I'm personally and we as a company are very happy that this is happening. So well done, organizers, to keeping the momentum and having it on an annual flow. Also, it's very good to see that we kept that flow also during these different times that we are currently living, and hence the topic of this call will try and to combine a bits of manufacturing and CMC, but also very much related to the antimicrobial resistance, or AMR. And the timing couldn't be better because I don't know if you are aware, but since last Wednesday until the end of today, this is actually a global antimicrobial resistance awareness week, which is pretty much a global thing, and many different companies and different organizations are trying to use that awareness week to raising the public knowledge and understanding of the... Guys, you need to be on mute, whoever is not. Thanks. So it's trying to increase the awareness of the AMR as a potentially rising pandemic. So the structure of this call, if I'm able to move the slides, yes I am. I will very briefly try to set the scene, and it is quite relevant to just reintroduce AMR. I'm sure many of you are familiar with it, but nevertheless, it's good to familiarize ourselves again. Then briefly over push and pull initiatives, and I will explain you when we come to that what that entails. And then the major part will be around the manufacturing, or CMC. CMC stands for Chemistry, Manufacturing and Control. It's a very pharma defined abbreviation, but it's actually everything that revolves around manufacturing and how you do it in a reproducible and responsible way, and also will a bit touch on the relevance of formulation. So if we are to set the scene, and I know you guys are from the field, but nevertheless, just to state the obvious, antibiotic resistance is an evolutionary thing. So the question, will a bacteria develop a resistance to any antibiotic? It's a generic question, and the answer is always yes, because resistance mechanisms are evolutionary ways how bacteria are fighting their enemies, and antibiotics are the enemies of the bacteria. Hence, they will try to do what is in their arsenal of mechanistic approaches to try to develop a resistance over any given agent. Of course, how we use them will have an impact on the dynamic of that process. And I pretty much like this slide. I know it's used quite often for a more layman's audience than you guys are, but nevertheless, it's very easy to understand how the resistance happens. We are, in a nutshell, with inappropriate use of antibiotics killing the normal bacteria, leaving the, let's say, the environment that bacteria are living in open for the resistant bacteria, which then have much more food and much more, let's say, nutrients for them available, because we kill the rest. And then they basically flourish, they grow, they multiply. And of course, they reach to a level when they become quite medically concerning. And that's not all, almost like on this marketing commercial. They can even, by some other molecular mechanism, let's say, translate and their resistant mechanisms to other bacteria. And by that, we have, transference of the problem across even within the species, but also across different species. And again, very well-known list and a very well-known abbreviation, ESKAPE. And I don't know if you can see the pointer, but basically this is the WHO priority pathogen list, which is quite similar to what the Center of Disease and Control from the US has. And European version is also very similar. But these 5 lines are pretty much the biggest problems, the bacteria that are causing the most problems out there, because they are quite difficult to treat, and they are a multi-drug bugs. And this abbreviation ESKAPE stands as a acronym or abbreviation for these bacteria. So Acinetobacter baumannii as a gram-negative bacteria is A, Pseudomonas is the P, Enterobacteriaceae are covering 2 bugs, Escherichia coli and Klebsiella, so this is the E and K, and then Enterococcus and Staphylococcus are the first 2. So these 6 bacteria, so 2 gram-positive and 4 gram-negatives, are causing over 90% of AMR problems in the world. And if people are unlucky and are picking up some of those, especially the gram-negative ones, there are some agents where literally nothing works. And all the antibiotics we have in our arsenal will simply not help. Because of that, there are many public health initiatives being run by different countries. So this is one of the very renowned one done by the UK government back in 2014, and many different audiences are citing those numbers. So by then, the estimate was that you have around 700,000 people dying annually from the infections caused by the multi-drug resistant bacteria. And they have predicted back then that if we don't do something as a global society, working together across governments, industries, academia, and each and every individual, that by 2050, the number of people dying annually from the AMR causes will rise up to 10 million. and will surpass the cancers. We are all very mobilized as a global society around oncology, whereas AMR, if this prognosis come true, can become the world #1 healthcare problem. As I will try to convey to you during this presentation, there are hopes and there are lights at the end of the tunnel that this will not be the case, but nevertheless, we must be aware all that this is a rising global problem that could have a very detrimental impact on our global society. And also, as you can see on a slide, it is quite global thing. Guys, please mute yourself. And it's spread across the different geographies. This is also what are the main causes. So there are, because this report was done and coordinated by Jim O'Neill, who used to be a chief economist for Goldman Sachs. And in a way, he used the economic vocabulary in trying to describe what we should do to fight antimicrobial resistance. And it's very simple. We should lower the demand and increase the supply. So very economic way of looking at things, and it's spot on. So on the demand part, I will just mention 3 things, which are one of the main culprits that we have a problem of antimicrobial resistance. #1, probably even #1 problem and the root cause is use of antibiotics in food industry in terms of animal farming and fish farming. So in agriculture and aquaculture, especially by breeding animals for food, where antibiotics are used not to treat infections, but as a biomass inducers. Also, one other element of causing the problem is, of course, use of antibiotics in human practice. And this is overuse, abuse of antibiotics, especially when they are not warranted. So if you are having some viral infection, you absolutely don't need an antibiotic. Antibiotics should be prescribed for the bacteria that are causing the infections, and many other prudent microbiological proofs are needed. So that's also very important root cause. And last but not least that I want to mention is the basics, and which is called sanitation and hygiene. Washing our hands and applying a basic standards in hygiene is doing a lot to help prevent resistance from occurring. And this is something which we all as individuals can do. So I did say that AMR is a global problem. It does require joint efforts across governments, industries, academia, but also starts from each and every one of us. So we can also contribute to the problem or to the solution. If we are applying and adhering to a basic hygiene rules, which also now we know during this current COVID-19 pandemic, that this is also one of the basic measures that one can do. We also can be very responsible patients and parents in how and when we use antibiotics when they are warranted. And also it's smart to pay a bit more attention on the food, what we eat, and try to minimize the industrialized, processed food as much as possible, because there's quite a lot of antibiotics there. So we all, as each and every individual, can contribute to that. And now what we can do to increase the supply, this is where I will focus the major part now on the call. The first part is basically increasing the amounts of drugs or discovering novel effective treatment options that will target some of those 6 culprits from the ESKAPE category, or some others as well on the list. And this is very well known in the press as a push initiative. So push initiatives are targeted where we want to increase the armamentarium, increase drugs that are offered to treat infections. And then the bottom part is related to the pool initiatives, which are more related to the business model. So if we are to start with the push part, somebody is not on mute. So on the push part is looking at what is in the pipeline. And I don't know if you can read, it's a sort of a bad quality slide, but apologies for that. But we have analyzed, or some of my and our colleagues, and published back last year in Nature Reviews, looking at the preclinical pipeline of all programs that are out there. And the good news is there is a lot of innovation in the field. And you can see that it's spread across different categories. But what I would like to draw your attention here is on this upper right part, where you can see that the majority of innovation is coming from small and medium-sized companies. Here, the definition for small and medium-sized companies is everything that is below 1,000, so 1,000 employees. So 81% of innovations and all these projects are coming from small and mid-sized companies, 12% for various academia, either universities or public institutes, and then only 3% are coming from large companies, which are defined by having more than 1,000 employees. And one of the reasons we have that picture is because of the pull part that I'm coming shortly to. And this is the broken business model of anti-infectives until recently, where, you know, whenever you discovered a new drug, your drug was shelved, put there for the last resort, which is absolutely medical right thing to do. And it's following the concept of antimicrobial stewardship, which is a great healthcare approach how to limit AMR. And antimicrobial stewardship is saying you always start treatment if you are not aware for what a specific resistant type is with older antibiotics, and then you gradually move up and reserve the newer ones for last line therapy. Hence, your volume will be very, very small. Hence, the revenues will be very, very small. Hence, the big guys completely left the field. And that's why you see only 3% of the companies working in the AMR are from the group of large companies. When you look at what are these assets addressing, they are addressing, as you can see, majority gram-negative bacteria, which is correct because these are the areas where we have the most problems currently, and it's most difficult bugs to treat. There is some broad spectrum programs, which is a combination of negative and positive, and then some that are directed to gram-positive, and then the rest is more specific of the above. But also what is very important, just look at the blue and greenish. There is 72% of assets are targeting green part, and green parts are new modalities, new class of compounds, new modes of actions, new targets. And the relevance of that is, if you remember what we said at the beginning, that resistance is an evolutionary thing. Whenever you are targeting new modalities, new class of compounds, you are slowing down the resistance development. And the good news is that over 2/3 of different projects are addressing new targets. Only a bit less than 20% are the so-called me-toos, which are addressing the old and existing, very well validated targets. And yes, those programs have a higher likelihood of developing resistance faster, but much more are targeting new targets. Also, there is a lot of money out there, both private, but also public-private partnerships as well, which are funding various small and mid-sized companies from the first slide, which are comprising 81% of the companies, and trying to help them during the discovery cycle. I'm privileged to be part of one of them, and that's a repair impact fund. And our mandate is that in the course of 5 years, we want to disseminate and basically distribute $165 million across projects and companies that are developing new drugs that are aimed at treating antimicrobial resistance. And you can see Carbax is from the US, it's close to or over $500 million. Then BARDA, it's a US thing. GARDP is a pan-European, like IMI as well. So there is a lot of money to be had. And just this summer, it's another fund that we, as a repair, among others, help to fund. And it's called AMR Action Fund. And here you have $1 billion available for the period of next 10 years with a mandate to try to bring 2 to 4 new antibiotics on the global market by 2030. This fund was done, and this is a great example what we can achieve when we work together. We as Repair are part of it, but not only us, it's more than 20 different companies have contributed $20 to $50 million each to this fund. Companies like Teva are part of it. Also quite a lot of Novo Nordisk is part of it, of course, due to the Repair link, but many other both biotech and big pharma companies that are not in the field of anti-infectives have contributed more as a philanthropic approach, $20 to $50 million each to construct that fund, which has a main focus to fund phase 2 and phase 3 clinical trials. When we come to the pool side of things, so I would say to conclude, push is looking quite good. There is a lot of money to be had to help companies develop projects across the value chain and come to the approval stage. But then comes the pull piece. So what happens when you get there? When you get your product approved, how you will be able to successfully launch it, and of course, as a company, enable a steady supply of a product and also recoup a part of your investment. So if we are looking at the anti-infective companies that are operating in the field, you can see a quite dark or quite gray picture. So these are, and this is quite old, quite old. It's a year-old slide, but things are even worse now. So it lists all the US biotech companies, which are here listed on the right-hand side, and some are more European, like Polyfor and Magrilia. But nevertheless, the companies that are exclusively working in the AMR field, on average, they lost 60% of their market value. These companies are listed on NASDAQ stock exchange in US. Even worse than that, there is on this slide only 4, but since I made this slide, there are 2 more companies that went back. So Melinta here is also bankrupt. So the companies that managed to get the product to the market, and then unfortunately, once they launched the product on the market, they simply went belly up. I can give you the story of Ekeogen, which is a company that bankrupted last year. It's a Bay Area, so West Coast US, California company that successfully developed and got approved one product that is active against carbapenem-resistant bacteria, especially relevant for the complicated urinary tract infections. So they are targeting top 3 names on this WHO priority list. The product name is plazomicin. They spent over $500 million over the course of last 10 years by developing it. And then when they got the product FDA, EMA approved, they managed to sell in the course of 1.5 years only 20 to 30,000 vials. Reason being, as I was trying to explain a bit earlier, is the antimicrobial stewardship and basically a medically justified approach that you keep those agents for the last treatment. And because of that, plazomicin was kept on shelf, was not prescribed to a patient, which was medically correct. But the company sold only 30,000 units and it went belly up. It basically bankrupt. So this business model where you pay for the anti-infective therapy, pay per the volume sold, doesn't work. And that's a reason why we are like to say that business model is broken. And that's the main reason why the big name companies have left the field. The good news is that now you can see from the AMR Action Fund that philanthropically companies are starting to put some money in, but also more importantly, governments have started to move. And I will at the very end of the presentation give you 3 examples, one that are in execution and one that is being discussed by politicians. And the country that is leading the way is United Kingdom. And they are really changed the complete business model of anti-infectives, and I will give you a brief explanation how that works in UK currently. So this describes the environment that we work in. So push working, pull completely broken. But pool part is now with some government interventions in countries that I mentioned. And I know that Germany is trying to do something now. So many countries will follow. And I'm hopeful that the business side of things and the pool side of things will also get improved over time. But the question is, and this is how this whole session is about, where does the manufacturing fit here? And I will give you two real examples and case studies. And they are true stories without any names to be disclosed. So this is a story which is some 20, a bit close to 25, but years ago. So it was a molecule that was discovered way back in time. It's a broad spectrum with a known mechanism of action. So this would be the blue part on the chart. And what happened here? So just briefly, the company had a compound that was very active. It was meeting all the in vitro and in vivo targets. And it was decided in the company to progress to the next stage. Whenever you are moving in your discovery effort to the next stage, you need more compounds, which is a common sense. So you need to make more compound. And when this company made new batch of a higher scale, this compound was completely inactive. Then this was confirmed in triplicates, both on the chemistry side and on the biology side, so on all the testing of activity. And unfortunately, how this works, it confirmed not the first, but the last. So it confirmed that basically molecule doesn't work, that this drug candidate is not a drug candidate, it's completely inactive molecule. So it was a complete project failure. The activities were stopped and project was killed. But the good news in this case, company, because it was a big pharma with enough money, they did perform detailed root cause analysis. And they identified solvent impurity as the main reason for it. What it meant basically that initial batch had a lot of solvent impurity in itself that wasn't captured in the specification. Nobody analyzed for that, so they didn't know. And that solvent impurity was killing the bug, not the compound itself. So this proves how manufacturing aspects are important. Second story is from last year. And it's from a small company that was basically formed as a university spin-out from a very well-renowned Western European university. The Biotech company was formed. They raised some serious seed capital, over 10 million euros. And they had a completely novel compound, novel mechanism of action. So the green part, which is great, with a narrow spectrum. They were also meeting all the in vitro and in vivo criteria. And then, as in previous story, they needed to move up the value chain. They needed more material. And they are then engaged some contract manufacturing organization, third parties, to make those. When the third parties started to manufacture the material, they couldn't reproduce anything that was written in the lab notebooks from the university. Of course, the postdoc and some others that were involved in that initial procedures left the university. So you had no, and the contact was lost in addition. So CMO eventually succeeded after complete reinvention and modification of the procedure, but that material was completely inactive. The same was confirmed by another third-party CMO. So basically the same outcome as in the first case. Here all activities were stopped. Project was killed because this was a small company. They didn't have even money to conduct any root cause analysis. The company is basically bankrupt and liquidated as we speak. So what are the key findings and learnings of these two real stories from big and small companies? And I would just draw your attention to the bottom part. It's a very critical, even though we are at an early stage of a discovery process, that we are adhering to a best possible quality attributes, either of starting material, of analytical methods and specification, because in the first example, if the solvent was part of the specification and solvent impurity, you would see that in your case, there was something fishy. Of course, you can ask the question, how was mass balance? achieved when they didn't catch that, and it's a very legitimate question to ask, but that also speaks for the quality of analytical methods. And overall, the second example is speaking about the quality of documentation. Many times, especially in smaller companies and academia, quality of documentation is not to the same level as in big pharma, and that can pose a big problem, because when people leave, people go different places, and if the documentation and the written part is deficient, you will have a problem when you need more material. So I know that, and also from experience and being in contact with gazillions of companies, especially in the discovery stage, they really hate GMP, and some of them consider it as a great mountain of paper. But in a way, GMP, of course, appropriate GMP principles and the GMP way of thinking to the discovery stage and early development is your project enabler, because it will protect things like that from happening. And it's on both process side and on the analytical side. How manufacturing is relevant is even more pronounced on the formulation side, because especially for these early stage companies, formulation is something they don't even consider as an item to be addressed. And now this was a survey done where over 100 different biotech companies participated, and they have been asked a very simple question: have you experienced a project failure or a delay due to formulation challenges? And you can see that a staggering number of 60% of participants said yes. And it either resulted that, you know, they in 10% of cases completely killed the project, or they had a significant delays. 52% said delay which was more than a year, and 38% project delay which was less than a year. So formulation is another underestimated aspect in basic discovery and early development processes, as well as the quality of documentation, analytical methods, and specification and recipes and whatnot. So we absolutely need to all be aware of that, and this is one very important consideration, how we can help fight the AMR challenge we have at our hand. So formulation, we should do it right the first time and from the very beginning, because it can help a lot. And last but not least, I would like to conclude with, and this is really one of my favorite quotes that I'm also using quite often, and it is from Dame Sally Davis. She used to be a Minister of Health and a chairperson for NHS services in UK, and one of the biggest advocates for AMR in the world. So she's in a way comparing COVID-19 with the AMR challenge, and is saying that COVID-19 is like dropping the lobster into a boiling water. You will instantly know that you have a problem. On the other hand, AMR is when you are gradually heating the water. So your lobster will not leave the water because you are gradually increasing it. You will cook the lobster at the end of the day, but when you see that you have a problem, will be too late. And I think it cannot describe the situation we are at hand so nicely as and so graphically and symbolically as it is. And because of that, the government changes that are happening, and again, UK leading the group, a lot contributed to Dame Sally Davis. So what they did, so UK saw that this linking of revenue with the volume is no good, and they were thinking, what else can we do? How we can design a reimbursement process that will work? And then they basically came up with a subscription model, so completely delinking value from volume, and then works in the following way. So when you submit for a registration, they will review your dossier, of course, give you approval or not, which is completely regulatory thing. But in parallel, they will do something which they call HTA, Health Technology Assessment. And that's where they are looking at a new drug and the medical benefit it offers to a patient and to a wider society. If they consider that new drug, so it's not applicable to all, it's applicable only to drugs that are addressing real and genuine unmet medical need, then they will give up to 10 million pounds per drug per year for a period of 10 years. So you can get cumulatively up to 100 million pounds over 10 years for just one drug that will offer an AMR solution. In the case of Ekeogen, they sold these 30,000 vials, and this was something like $2-3 million altogether. Here, for one market, UK, not US, which is a smaller market, you can get up to 100 million pounds over 10 years, irrespective of the amount of units you will sell. If you sell one vial or one capsule, one tablet, or tens of thousands or millions, still you will be rewarded by the medical value. Something similar is being executed in Sweden, a little bit different, but very similar. And then in the US, there's currently a new law that's being proposed by the bipartisan activity, believe it or not, of Republican and Democrats working together. It's currently in a stall due to the known challenges in US, but follows exactly the same logic of UK with this market rewards, which can be up to $3.5 billion over a period of 10 years. So with that, it's clearly seen that the environment is changing to a positive, that there is light at the end of the tunnel, and that with this, I'm convinced personally, and we as an AMR society, that probably the number that I quoted for 2050, if this continues, will not happen. And with that, I would like to end this run through the quite a few slides. Thanks.
I need to unmute myself. Thank you so much. Thank you, Sasha. Thank you for this presentation and for this great overview. And I think especially for what I would say, you know, warning us and maybe announcing and something that we see both the danger, I would say, that comes from the AMR, but also this policy changes and potential really to reorganize how do we address healthcare and how do we plan for healthcare in the future, which is very hopeful. Also, I would like to really thank you for you know, singling out a couple of examples of you know, importance of thinking about the manufacturing process and thinking about formulation process very early on in development. Of course, scale up and others is one of the big dangers. I think also one of the things that you singled out that you sort of jumped up to me as somebody who comes from the university is there, you know, this one case that you mentioned where there was actually a collaboration with the university, and you know, and issues in transfer of the results to the industry setting. And then also you mentioned, you know, like postdoc left and the results couldn't be traced back and so on. And you know, this of course happens, and I would like to ask you, and I maybe ask your opinion, because you know, how would you, if you could optimize, you know, the collaboration with universities or the collaboration with the academic sector, the way you could possibly optimize, you know, whether it's the formulation or the manufacturing process, you know, how would you do it? You know, and I'm asking for your comment now because I know you won't be able to join us for the discussions. So maybe jumping in, and if you could please share your thoughts on this topic as well, you know, on the technology transfer and your academia industry collaboration. And I know that Xellia is very active working with universities here in Croatia and probably across the world.
Yeah, it's the real question. You nailed nailed it in the head. So yes, universities are places where majority of world best science is happening, and that's undisputable. Where, you know, the collaboration would benefit and where universities would benefit from the industries, as I tried to point out on the documentation piece, and on the fact that sometimes it's good to repeat. So yes, you can have a great result initially, but you know, if you didn't repeat it few times, in a way, you shouldn't claim you have a good result. I mean, in industry, we do that as a normal practice. And even when we have something which I know some of the people on the call are very familiar with, like OS, out of specification results, their first rule is to repeat. And this level of repetibility and being very cognizant about the importance of that, it's something which universities, and I'm not saying in Croatia, this is very global and universal. And as you said, we are involved either through Xellia or either through repair by many, many different, even Ivy League universities. And it's very common that the quality of documentation is very poor. You know, the procedures are not written well. So how it was done? It was done by the Mark. Mark knows it. But who is Mark? Mark can lead. And Mark, of course, has notes which are, if given to anyone, unreproducible. And we stumbled on that example, which I had as a case study number 2 more than once. So I would say understanding the importance of documentation, understanding the importance of quality attributes, understanding the importance of analysis, and repeating the good result is of paramount importance. Because you can have an outlier. Sometimes good result is not a result, it's something which is outlier. And if you didn't repeat it, you don't know it. And also there is a lot of scientific papers out there which are struggling with that as well.
Yeah, there was actually, I think I'm forgetting the numbers now, but you know, some pharma company did this analysis of how many results could be repeated, and yeah, the results weren't too great actually for the academic sector, and again, it was a global analysis. It wasn't linked to any single country. I don't see any questions from the audience, so maybe, you know, just to ask maybe one more thing, you know, apart from this, you know, are there any other things specifically looking for, I would say, academic sector, or maybe how we can better cooperate? Any final thoughts or messages?
I mean, always have the end game in mind. You know, in every collaboration with the industry, there always need to be a solution to a real problem, not to the perceived problem, but to the real problem. And this is also like the UK solution to the broken business model of antibiotics, is exactly addressing that. So not any drug will get this subscription of up to 10 million pounds, only the ones that are fixing the real and genuine problem. The same means for any other, you know, academia activities, research, discoveries in any field. Doesn't need to be AMR or pharmaceuticals or life science. Can be in any other areas of work. It needs to provide a solution to a real problem. Not to the problem we invented and then we found a solution to it, but to the real practical problem. And this is how and when any industry will jump in.
Thank you so much. Thank you for this insights, and thank you also for the patience with the technical difficulties that we had in the end. I know that you won't be able to stay until the discussion. So really, I really do want to you know, tell you how much I appreciate that you have been here with us and being able to share the insights that we have. As I still don't see any questions from the chat, let us move to the next presentation step, and I hope that the speaker is also here with us. So the next speaker is Sandra Miočić.
Hello, everyone. I'm here.
So hello, Sandra. Good that you are here with us. So Sandra is a principal scientist and formulation leader from PLIVA Croatia, which is part of a global company, Teva. And she is going to talk to us about some specific issues, or I would say important points that need to be optimized in order to have you know, to have the functional drugs, and basically also comment on the innovation aspect. So please, Sandra, go ahead.
Thank you for the introduction. So in today's presentation, we will cover visible particles control with a specific emphasis on challenges with injectable suspension drug products. So we all know that visible particles is a hot topic in the industry, and that regulatory requirements around the presence of visible particles in injectables are getting more stricter. And we can see that around 48% of all the injectable drug products recalls is related to the presence of visible particles. So injecting visible particles is related to different patient risks. It can be, this level of risk depends, of course, on route administration, with intravenous route having the most risk, and also can be related to embolism, granulomas, inflammation, and infection. However, for some other routes of administration, intramuscular and subcutaneous, this level of risk is much lower. So just to briefly remind us what are visible particles, according to USP definition, these are mobile undissolved matter other than air bubbles that are unintentionally present in injectables. And regulatory and compendial requirement, both for USP, EP, and other pharmacopoeia, is that injectable products should be essentially free or practically free from any visible particles. Due to this essentially free requirement, it is really hard to understand what are the real acceptance criteria when developing injectable drug products. Also, the size of visible particles that can be seen by the human eye is not clearly defined. So it can be in the range of 50 microns to 150 microns, depending on the variability between operators, and also the probability of detection of visible particles gets higher with increasing particle size. So just to briefly go over the sources of contamination with visible particles of drug products. So there are 5 main sources. So particles can be related to the process, to the environment, personnel, packaging materials, of course, formulation, API, excipients, and also can be generated during the shelf life as a result of interaction between the product and the primary packaging material. And they are classified in the 3 main categories. These are the extrinsic particles that are the most critical. These are the particles that come outside of our process and are unknown and uncontrolled, and their bioburden is unknown. So it is important that they be eliminated from the product. Other category are intrinsic particles. They have a lower level of risks. These are particles that are part of our product or process. They can be generated by the manufacturing equipment or by the primary packaging materials. And such particles should be controlled and monitored in the product. Also, the particles of lowest risk are inherent particles. These are particles part of the formulation. They can be, for example, in suspension, some aggregates of active pharmaceutical ingredients, some opalescent excipients or protein aggregates. And for them, it is important that those particles are characterized and monitored over the shelf life of the product. So in reality, we have this balance between the regulatory requirements and industry practice. So from the regulatory side, it is required that we have 100% units in inspection, that we have 0 defects, that everything is 100% particle free, and all the particles are eliminated. And in the reality, it is unrealistic to have a completely particle-free process, since particles are everywhere. And also we have this probabilistic nature of inspection process, which depends on the inspector, and they can detect different sizes or not detect different sizes of visible particles. So to aid the industry, Pharmacopoeia came up with a statistical approach. So it is required after that you do 100% visual inspections of the batch, that you take additional sample where sampling plan and acceptance criteria are defined based ANSI and ISO standard. And inspect these samples. In this way, it is easier for the industry, because some more comprehensive acceptance criteria can be defined for different products. So what is AQL, which is mentioned in this ANSI standard? So it is basically acceptable quality limit. It is, let's say, a worst case scenario batch that would still be accepted as of adequate quality. And it's a measure of inspection, visual inspection process integrity. Based on the USP requirements, these acceptance criteria are defined according to the batch size of the product and pre-described inspection level. So for standard products, let's say a transparent solution, general inspection level 2 is used. And based on this and specific sampling plans and required acceptance quality limit of 0.65, you can determine the needed sample size and acceptance criteria. Also, for products like lyophilized products or suspensions, where you need to apply destructive methods for testing, Pharmacopoeia allows reduced sampling according to S3 special inspection levels. So, for example, in this case, for a standard product, you would need to analyze 315 units, and acceptance criteria is to have 0 visible particles in these units for certain batch size. And for suspensions and freeze-dry products, it's 20 units should have 0 particles. So we mentioned that this visual inspection and particles are challenging even for the transparent solutions, but what about difficult to inspect injectables? These include opaque solutions, colored solutions, white suspensions, emulsions, and lyophilized cakes, and also other types of primary packaging where inspectors during visual inspection cannot detect the presence of particles. So for such products, supplemental destructive testing or inspection is needed. And these products are of higher risk for batch rejection, because in case some of the, during this destructive testing, you find some of the visible particles, you cannot reinspect the whole batch, but you need to reject the batch in the end. So for such products, specific methods for destructive inspection need to be designed. So for powders and cakes, you reconstitute and inspect. For deeply colored solutions, they are filtered and filters examined for presence of particles. And for emulsions and suspensions, basically a solvent is used which dissolves API, and then the resulting solution is analyzed for presence of particles. And for non-transparent containers, it is the easiest, so the contents is transferred to the clear container and inspected. So for the suspension products, we have specific challenges. So here we have comparison of a manufacturing process of one standard transparent solution and suspension. So for solutions, the standard procedure is to do the compounding, dissolving API, and do sterilizing filtration when 0.22 micron filters, which is also done at the point of filling into the containers. So we can see that for the transparent solutions, it is easy to get rid of any visible particles with this sterile filtration step. And for suspensions, the manufacturing process is much more complex. We have vehicle preparation, API suspending, then we can have multiple aseptic high energy steps in the process, particle size reduction with high shear mixing or wet milling, and then filling into vials. And for both of these types of product, the primary packaging containers have the low risk for the particles, because they can be, if we use ready to sterilize packaging, they can be washed, sterilized, depyrogenized at site. And for ready to use primary packaging, then the supplier needs to control the absence of visible particle. So as we compare these most critical parts of the process for the visible particles, we can see that for the suspension, from the beginning of the process until the end, we have critical process steps that can introduce visible particles. And of course, for the suspensions, we cannot apply filtration, because we would then filter out the active pharmaceutical ingredient. Also, we see in the industry that it's getting more and more recognized that visible particles in raw materials is also critical for some types of product. So we see guidelines getting from some excipient suppliers and active pharmaceutical suppliers, where they prescribe some guidances for the control and monitoring of these visible particles. And they call them technically unavoidable particles, which are generally present. And it's important to note that they have some of their own control criteria and requirements, which are not in line with requirements for suspension drug products. Also, we can see that they are not really harmonized with final drug product manufacturing, as they note that these particles, for example, for injectable drug products, would be filtered out and are not then anymore critical, but for suspension products and some other, let's say, products based on polymers where we cannot readily use filter steps, it is not then applicable. Now we will get into some case studies and some challenges that we encountered in development of suspension products with visible particles. So we're working on one freeze-dry lyophilized powder for injectable suspension, which is in a suspension form after a reconstitution. It is for intramuscular administration. Bulk suspension has about 10% API solid content, and particle size in D90 is 10 to 15 microns. So we manufactured one small pilot batch, 100% visual inspection of lyo cakes passed, and everything was okay. And then we proceeded to the supplemental testing. So for suspension and lyo products, it is mandatory to apply destructive method. So sample is dissolved in organic solvent acid mixture and then filtered out to a black and white filter paper, so you can see all the particles present. Sample size was defined based on the ANSI requirements. So our acceptance criteria was to have 0 vials with visible particles from 20 vials sampled. So after the analysis, unfortunately, we realized that in this product, we have black compact particles. And not just that, there are also present white fiber-like particles in the product. So this was then a doubled problem. So immediately investigation was opened, and we started with dark particles, black particles. First, the particles were isolated. They were analyzed by Raman microscope, ACM, with multiple methods, and it was defined that these are black compact particles up to 200 microns in size, with elements of carbon, oxygen, chloride, with no Raman spectra, and with characteristic spectra by MALDI TOF MS. And in the parallel, there was manufacturing equipment inspection in the production, where after they dismantled the complete compounding system, found one damaged rubber gasket in the high shear mixer. This gasket was then characterized, and the spectrum was matching the visible particles found in our samples. So this deviation was, let's say, pretty quickly resolved. And corrective and preventive action put in place were inspection of the gaskets after each batch, more frequent changes of gaskets, because as mentioned in suspensions, these are pretty high energy processes, and there is a high risk of wear and tear of the equipment and the batch contamination. So after this, we could move on to investigation for the white fibers. So here we started with API inspection. So API sampled into particle-free containers was dissolved in the organic solvent and filtered into white and black filters. And soon here we identified white fibers in the starting raw material in API up to 200 microns in size. We immediately informed the AP supplier, and they, by their inspection of manufacturing process, they noted that they use a centrifuge bag for API filtration, which could be a possible cause, as they could shed particles during their process. We have took a sample of this centrifuge bag and indeed confirmed it as a source of visible particles in our product. So the action after this would be to work with the supplier that they eliminate particles from API, to set a specification for visible particles in API, which would even have more strict AQL of 0.4, so that we are sure that we do not have out of spec in our drug product. But of course, this was not so simple. This would mean that the API supplier needed to change the plant, need to move the process to the plant with environmental monitoring, or even to the sterile plant. So there was a big impact on our timelines, and also such sterile or particle-free API would be even 20 times, have a 20 times higher price than the regular API grade that we were using due to these specific requirements. Therefore, we needed a quick solution to move on with the project. So we were thinking, is there any way we could filter out the visible particle but not affect our suspension? So our suspension had, let's say, the largest particles were between 50 and 90 microns. As a first approach, we tried some standard depth filtration filters, single use, with higher pore size of up to 100 microns. But as we expected, there were issues with filter clogging because these depth filters remove much more smaller particles than their pore size. So they were in fact removing API particles. So as the next idea, we thought about some stainless steel mesh that we could implement in the process, and found these sintered stainless steel filter cartridges and we selected this particle removal rating. So they remove 98% of particles larger than 70 microns and 1% of particles larger than 105 microns. So for us, this is the great range because knowing the visible particle size, they would then remove visible particles. So this was the first setup for this process in our lab. This is a filter in the filter housing, and after filtration, we analyzed the resulting suspension, and we get really great results. There was no effect on particle size distribution, no change in assay, and the resulting sample was free from visible particles. So we needed to move really quickly and implement this into production. So production was at the beginning reluctant for this because they never used anything like this in sterile production. And we needed to introduce this filter into a aseptic process. So from the materials of constructions, it was acceptable for parental manufacturing. Also, it could be sterilized by autoclave, and it could be easily connected to the suspension transfer line via septic connectors. Regarding the process parameters optimization, we needed to run this transfer at really high rate to prevent any API sedimentation, acid loss in this filter housing, and no stoppages during transfer were allowed to prevent filter clogging. Also, to have a better yield, we added one additional nitrogen purging step to minimize losses, so the yield in the end was very good. So in the end, this filter was successfully implemented. We manufactured a clinical batch and which passed all the AQL requirements for visible particles. So we have successfully removed these visible particles, these white fibers that were coming from the API. And this is just the last slide, which summarizes all the risks for visible particles in suspensions. So as mentioned, these technically unavoidable particles are allowed by API suppliers and are not aligned with compendial requirements. For sterile APIs, this risk is lower. However, if we have sterile API, it doesn't mean it is particle-free. So it also needs to be controlled, and also API packaging could be a source of contamination. Another very important point is the process. So in suspension manufacturing, we have multiple high energy process steps, which are high risk for equipment wear and tear. These are complex processes. We have more tubing, more connectors, more gaskets, more contact parts, which are potential source of contamination. And also it needs to be cared also for single-use systems and API dispensing step, and for filling that there are no further particles generated by these steps. Excipients are of lower risk because usually in the process we can implement some clarifying filtration step and remove potential visible particles coming from excipients. But for some products, as for viscous polymer solutions, it can be challenging, and filters with large pores need to be used. And for the containers, we have similar risks as for transparent solutions. So some takeaway messages how to reduce this risk. Close collaboration with API supplier is needed early on into the development. For critical products, we need to set specifications and use particle-free packaging. And for process, it needs to be equipment materials of constructions and design need to be such to minimize particles generation. Everything from the contact materials needs to be washed pre-use. System needs to be closed as much as possible, and use the filters whenever possible, closest to the point of filling as possible, and use large pore mesh filters. And as in our example, it was a simple solution, but in the end, it resulted in no delay in timelines and no additional budget to the product. So thank you all for attention.
Thank you, Sandra. Thank you very much for this presentation. And I think there are quite a lot of participants in the audience that come from the academic community, and we heard also today that earlier today that it's so very important to actually solve the real problems. And I think it was a really great opportunity that you shared this very, I would say, real and very practical problem that lots of drug or manufacturer face, and different ways of I would say addressing it, and it really is complex. So there is the supply chain control aspect, but there is this also quality control aspect, and then jumping in and really doing detective work, and really trying to solve the problem in a short time span. And I'm really hoping that we can touch upon it more in our discussions, and other, to look maybe for ways for solving it, or maybe look for ways how we could address this as a part of this industry academia collaboration. We will now move to the next presentation, and as I don't see specific questions from the audience, we will then leave the questions for the final discussion round. So with that in mind, I would like to announce our next speaker, which is Maša Safundžić Kučuk, who is R&D Director for Jadran Galenski Laboratories. Maša also has very extensive experience in pharma industry, and
that regulatory required.
And also, I would say, so we are very happy to have her with us and to talk about Jadran Galenski activities in the space of generic medicine. So Maša, please go ahead. I know that you're sharing the slide. I hope you're also unmuted and can start your presentation.
Okay, thank you, Latka, for the introduction, and hello, everybody. I would like to thank everyone who joined today, and to be honest, it's quite an impressive number of the attendees. And also special thanks to the organizers for inviting me again to speak on this winter school. As Sasha said, it has become a tradition. And I'm especially glad to be here, as this event has encouraged collaborations between participants and JGL in many ways. And I really hope that in future winter schools, I'll be able to share more details about those collaborations. But today I will talk to you about how you can make innovation in generic industry, because it needs innovation. There are data that say that since mid-90s, only 85 to 90% of all new drugs provide few or no clinical advantages for patients, or in another percentage, that 11% of the drugs that have been approved between 74 and 94, only 11% are therapeutically and pharmacologically innovative. So also generic industry needs some innovation. I usually start with definitions, so I'll start also today, just to have some base definitions about what I will talk about. So, originator is the first drug that has came to market, is a new chemical entity. Its efficacy, safety, and quality data are based on clinical studies. Then on the other part is a generic, whose authorization is based on efficacy and safety data from studies on the authorized medicines. You need to show bioequivalence studies or to achieve a bio-waiver. That means that you can skip the clinical studies, and you really need to show the Q1, 2, and 3 sameness. Q1, Q2, and Q3 are, it's called in pharma industry levels of sameness. Q1 is that you have the same ingredients. Q2, it means that you have the same ingredients in the same levels. And Q3 means that you have everything the same and that it behaves the same. The quality is the same, the rheology and other parameters. Those are traditional generics. And now we are starting to see value-added medicines or new therapeutic entities or hybrids or super generics or added value generics. There are a lot of names that mean the same. things, and those are the known medicines that are based on known molecules that address healthcare needs and deliver relevant improvements for patients, healthcare professionals, and payers. So we can also call them generics with a twist or something like that. How does the process go from when the originator comes to the market? So the originator is the first one that is approved for the market release. The efficacy, safety, and quality data are based on the clinical studies. It's a lengthy and costly process, so we gave them a legal monopoly, patents, that give originators legal monopoly. In return, they need to disclose their invention. Patented period can be up to 20 years, and that means that only the patent owner can sell the patented product. 20 years can seem as a long time, but if you spend 10 years in clinical studies, that time goes really quick. So there is another way of, not another way, but parallel way of exclusivity that are given to originators. It's called data and market exclusivity. And that means that no one can refer to preclinical and clinical data of the originator in 8 to 11 years after obtaining a market authorization, nor to sell such generic. And also in Europe, there is a thing called a supplementary protection certificate that comes into force after the patent expires. It's up to 5.5 years, and it's a compensation for the period elapsed between the filing of the patent and the market authorization. Somewhere in that time, generic drugs come into play. Those are products that are identical and bioequivalent to the originator drug in terms of dose, form, route, and method of administration, safety, and quality. And their price is lower because there are no costs associated with clinical studies. In parallel or after generics, there can also come added value generics that are innovative medicines based on known molecules that address, as I said, healthcare needs and deliver relevant improvements for patients, healthcare professionals, or payers. And which are those improvements? I will talk in next few slides. So how can we and why should we improve generics? Sorry. On the market, there is quite big global healthcare inefficiency. And three top leading causes of this inefficiency is that we are not using enough of generic products, or that we are using substandard medicines and inappropriate or ineffective use of medicines. So we should improve generics. And how we can do that? We could improve clinical profile of a generic drug, and that requires very in-depth understanding on how a particular medicine works. In this case, the objective is to deliver the right drug in the right dose at the right time and at the right dosage form with the right drug release performance. It really needs you to understand very good the clinical pharmacology of the drug and what can be done to improve that. Then also you can facilitate the use with better product design. Quality in the hand of the patient is the requirement for develop the generic product, but this is one step further. This is a definition of a product quality as a quality in the hand of the patient, and the patient is using that product as it should. Because the patient is important for the therapeutic outcome of therapy as the drug product itself. And we need to design this product with the patient in mind in order to increase the compliance and adherence to the therapy. Because it's really a big problem that not all patients are using that medicine as they should. And here you have one estimation that for every 100 prescription written, not all are taken out of the pharmacy, not all are taken properly, and just 15 to 20% are refilled as prescribed. So patient adherence and compliance to the therapy is a very, very big issue today. Especially if you look from the point of view of a healthcare system. We are paying for those medicines and they are not being used as they should. We can also improve generics by increasing the manufacturing efficacy and reducing the costs of good by producing more in better and more efficient way. By cutting out the costs that are not linked to the API, excipients, or production. How we can even find the candidates for enhancement. So for me, that I come from a company, the usual way would be that we look at existing portfolio of our products with external mindset. That means that we have some advisory boards with doctors, pharmacists, and patient representatives. And this is really the way how one of our biggest products came into the idea, the seawater with API. We saw that, and in discussion with pediatrics, we saw that they are diluting our medicine with seawater, and then the idea of a new product has come to mind. Or you can use nowadays big data and digital technologies that allow you to go through large amounts of data like patient records, clinical trial data, publications, and see if there is a field or a need that is unmet. Or you can have a proactive approach by asking health authorities, bodies, doctors, patients, what are the needs, and what are the current problem with current medicines. Maybe, for example, we saw that in eye drops, the prolonged use of preservatives, it's not beneficiary. So now we are trying to take out the preservative from the eye drops, especially the ones that are used every day. And now a little bit more in depth that you can think about what can be and how value-added medicine or generic can be designed. You can think about reformulation of the existing product, repositioning, or a combination of some kind. So changes in dosage form can be, an example would be if you have a powder antibiotic for pediatric patients, you need water to reconstitute that product, and afterwards you need to put it in refrigerator conditions. Not all have access to clean water or refrigerated conditions to keep the medicine. Maybe it would be better to have an orally dissolvable tablet. It's easier to use, it's more stable, you don't need the refrigerator, and it's better for the patient that don't have such conditions. Or you can do something with the strength of the current medicine. You can lower or making lower or higher concentration of the API to lower the adverse effect or to get a new indication. Examples would be bimatoprost solution. If you look at classical eye drops, it's Lumigan with 0.0%. For treating glaucoma, but Latisse 0.03% is for growth of the eyelashes that are lost due to chemotherapy or cosmetic reasons or something like this. So they upped the dose in a new version. Or loteprednol It's 0.5 as Lotemax is a medicine for post-cataract surgery patients, but they found if they lower the dose in half and go to 0.25%, this is a fantastic candidate for dry eye disease, and EU Swiss now in last few months has received an FDA approval. Or you can change the route of administration. Midazolam is a medicine for sedation. It was usually used orally or intravenously, but the bioavailability then is 30%. And now they are looking into nasal route with better bioavailability for the patients. Very popular today are drug and drug combination. They are important when patients need to take multiple medicines several times a day. You know, you have mothers, grandmothers, or other relatives that have an array of medicines that they need to take at multiple times a day, and they usually forget something. So those are fixed combination products, and when they are combined, they can be tablets or drops. They are much easier to adhere to the treatment and not to skip important ones. There are also quite exciting examples of combining application devices with known molecules. There's Abilify MySite, a digital system that has aripiprazole tablets that have ingestible sensor in them. So the patient ingests them. They have a patch sensor that looks senses when the medicine passes through the bloodstream, and it's connected with a smartphone app. It's and that monitors ingestion over time, which is especially important for patients where you don't want them to miss their medication. Or an example of new indication or patient group, the most popular is sildenafil that was designed for engine, but is most popular drug for erectile dysfunction. That means for this type of change, you take the existing data or you generate the new data and find the new use or new population for this medicine. It's called finding hidden treasures in old drugs. So as you can see, there are numerous examples for each option here listed. And it's really a good way of innovation because you reinvent the old and not just new. So what are the R&D efforts in developing such medicines? Well, the amount of data that needs to be generated is very different from that is required for standard generic products. It's usually more, the approach is more similar to new drug entity, but because you usually need clinical studies, because to better understand how the body handles the value-added generics, to understand the effect of the drug on the body in healthy volunteers or in patient, and to monitor patient experience studies in the post-marketing setting. There is not really one approach fits all. For each product, you need to tailor the development approach, and that approach really will depend on the modification that you want to make to the medicine. But one thing is especially important here is that you know your legal basis or how you will market how you will register your product very, very early on. They are interrelated. First thing, you need to talk to the regulatory experts and to see what the strategy will be, how to put this product on the market. In Europe, you can go to the route of hybrid application or fixed dose combination. It really depends on what the change is. I won't go now into these details about how each procedure is looking at, what set of data is needed, but it's really, really important that you that as soon as you have the idea, you start collaborating with the regulatory affairs specialists, because you will need, in some point of time, it's more likely to, yes than no, that you will need a clinical development program to collect new clinical data. And when you compare length and cost of developing a generic and new entities, for example, generics, it's usually said that you need 3 to 4 years to develop a generic and that the cost is somewhere around $1 million. But for the new entity is 10 plus years sometimes, and the cost is very, very high, is difficult even to estimate some sort of average. Value-added generics, they are somewhere in between. As I said, depending on the change that are doing, the cost will go up or down. But it's really an exciting way of finding new use for the existing molecules, because I said the numbers, not all new drugs that come to market are innovative. FDA made estimation that only 34 up from 218 new drugs that they approved from 78 to 98 were judged as important therapeutic gains. It's really a low number. So this is why we really need to look into existing molecules and to find a new use for them. And that would be really the end of my presentation. I really hope that you will, that maybe someone from all of you participating in this winter school will look a little bit in closer look existing molecules. So Vlatka, I'm finished, and next presenter can start sharing the screen.
Okay. Maša, thank you so much. Thank you for this great presentation. We will leave the questions for the final Q&A, and I do want to stress this to all participants. Please do ask questions in the chat. So even even though we started a little bit late, we will find a time for the final Q&A session. And please, you can address of any of the speakers, or you can ask a general question, ask it now in the chat, and then I will be able to repeat it for you. And I'm really looking forward for this final presentation, and then for the discussion that will follow. So for the next presentation and for the final speaker today, we have Professor Anita Hafner, who is comes from the University of Zagreb, Faculty of Pharmacy and Biochemistry, so one of the co-organizers of the school and of today's event. And Professor Hafner is a lead researcher on what is the installation grant? I was thinking establishment, but it's the installation grant that was supported by the National Science Foundation, and I believe also involves some collaboration with industry. So we are very much looking forward to your presentation about this project. Please go ahead.
I hope you see the presentation.
Yes.
Okay, so thank you, Dr. Petrović, for your kind introduction. And also, I would like to thank the organizers for inviting me to take part in this exciting event. I'm very pleased to present an example of academia-industry collaboration in the field of development of innovative nasal drug delivery systems. And within the project entitled optimization of nasal deposition pattern of sprayable in situ gelling, and the powder drug delivery systems. As you said, it is funded by Croatian Science Foundation. You can see here the list of participating investigators, among which there are 2 PhD students, and also the list of collaborators, including experts from our industrial partner JGL. At the beginning of this presentation, I would like to present briefly the advantages of nasal drug delivery, which is in the focus of our project. So, nasal drug administration is the first choice in the treatment of diseases of the nasal mucosa, while at the same time, it presents a very interesting and very attractive alternative route to systemic drug delivery. This is mostly due to relatively large surface area, high vascularization and permeability, and low enzymatic activity of the nasal mucosa. Here it should also be emphasized that there is a possibility of direct drug delivery, of nasally administered drug to the central nervous system, and mostly via the olfactory nerve, presenting actually brain-targeted drug delivery. Well, all these possibilities of nasal drug administration are closely related to the characteristics of the nasal cavity and the mucosa. Thus, in order to ensure local drug effect or systemic availability of drug, it should be delivered to the turbinate region of the nasal cavity. And in order to ensure CNS drug delivery, olfactory region should be targeted. As you can see here, this region is innervated by olfactory nerve, with the neurons being directly exposed to the external environment. You can see here also the trigeminal nerve that can contribute to the mentioned pathway of drug delivery. Well, all these targeted regions are located beyond the nasal valve, which represents the narrowest part of the nasal cavity, and thus makes it difficult to deliver drug to these targeted regions. And another obstacle that should be mentioned is the mucociliary clearance that shortens the retention time of the drug at the nasal mucosa. So actually this brings us to the aim of the project, which is to develop sprayable nasal drug delivery systems with optimized deposition profile and optimized biopharmaceutical properties, of course, in relation to drug therapeutic effect. And within this project, we are working on the development of in situ gelling and powder delivery systems that are able to prolong retention and provide optimized drug release at the nasal mucosa. So let me briefly present in situ gelling systems. These are actually polymer dispersions containing active pharmaceutical ingredient that can be, of course, easily sprayed as a liquid formulation to the nasal cavity. These systems turn into gel in contact with nasal mucosa, ensuring prolonged retention at the site of administration. This salt gel transition can be triggered, or better to say is triggered, by physiological factors like change in pH or temperature or presence of ions. You can see here the schematic presentation of the gelation of pectin polymer chains in the presence of calcium ions. And this described rapid gelation reduces the postnasal drip, anterior leakage, and mucociliary clearance of the administered drug. In case of in situ gelling systems, formulation parameters that may be fine-tuned in relation to product characteristics include the selection of polymer and polymer drug concentration, as well as surfactant concentration in case of in situ gelling suspensions. As for the nasal powders, we prepared them as the carriers of active pharmaceutical ingredient by spray drying technique, which is single-step, easy to scale up method for the preparation of powder drug delivery systems. You can see here the schematic presentation of spray drying process, including atomization of the feed solution, drying of the droplets in the drying chamber and in the stream of the heated gas, and finally separation of the dried product. Well, in this process, apart from formulation parameters, also the process parameters may be fine-tuned in relation to product characteristics. And these include, for example, feed flow rate, compressed air flow, or inlet air temperature. As for model drugs we use, we actually started with fluticasone propionate. It is the drug with local effect used in the treatment of allergic rhinitis, rhinosinusitis, and nasal polyps. And currently, we are working with dexamethasone sodium phosphate, and we are trying to target olfactory region. Well, namely, very recently, nasal delivery of glucocorticoids has been proposed as effective strategy to control neuroinflammation processes in patients with severe COVID-19, and thus avoiding problems related to systemic application of high glucocorticoid doses. Targeting olfactory region could also be interesting in the treatment of anosmia and hyposmia frequently seen in these patients. So this is really an interesting and hot research topic. And also we plan to work with donepezil, which is a drug used in the treatment of Alzheimer's disease. Development of in situ gelling and powder systems is actually a very, very complex task and requires diverse considerations and characterization procedures to screen the therapeutic potential of formulation candidates. So in this regard, it is very important to elucidate the influence of formulation, process, and administration parameters on, first of all, sprayability properties, then behavior in the contact with nasal mucosa, and deposition profile. On this slide, factors related to sprayability are droplet or particle size distribution, as well as rheological or flow properties. While the behavior of the system in the contact with nasal mucosa may be screened by mucoadhesive or swelling studies, in vitro drug release studies, and biocompatibility and permeability evaluation. Our group is especially focused on the deposition profile, since the bright deposition pattern within the nasal cavity is the prerequisite for attaining the therapeutic effect. So to solve this complex situation, we use statistical design of experiments in development of nasal formulation, and we have mathematician support in determination of experimental design and analysis of obtained results. And you can see here an example of development of nasal in situ gelling fluticasone propionate suspension involving collaboration with our industrial partner, JGL, that actually shares the interest in situ gelling formulations. In this table, you can see parameters, both formulation and administration, and their levels included in experimental design that resulted in 42 samples and 10 replicas that were analyzed in terms of key responses, including zero shear viscosity, droplet size distribution, spray cone angle, and deposition profile. So our partner JGL support was provided through exchange of knowledge and experience in reaching very important starting milestones, including selection of the formulation components and proper spraying device. This is very important since spray cone angle being selected as one of the key responses was recognized as a critical parameter related to the efficiency of drug delivery beyond the nasal valve. And the collaboration with JGL was here very important due to their considerable expertise in the development and characterization of nasal sprays. Furthermore, JGL provided access to highly sophisticated equipment that needs to be used in development of nasal sprays, which includes a Lasso diffraction system that is used to determine droplet size distribution of spray formulation. It was very important in the early phase of our studies to assess if the obtained droplet size distribution met current requirements for localized nasal delivery. I would like to emphasize here that in this regard, PhD student was hosted and supervised by collaborators from JGL who have long-term experience in spray pattern and spray droplet size distribution determination. And as I mentioned before, one of the biggest challenges in this project is in vitro evaluation of a formulation nasal deposition profile. So as previously emphasized, such characterization is really very important for the prediction of performance and therapeutic potential of a nasal delivery system and may be considered for implementation in industrial setting, maybe presenting one of the potential results of this collaboration. So in our studies, we use commercially available nasal cavity silicone model connected to the respiratory pump in order to simulate breathing conditions. And in order to visualize the spray deposition, we use the SAR gel water indicating paste that, as you can see, turns purple in contact with water. We also derived regression model showing the influence of both formulation and administration parameters on the turbinate deposition. You can see here the concentration of drug, polymers, surfactants, angle of administration, and inspiratory airflow together with their interactions. Also, we made a step forward in this regard. In collaboration with Sisters of Charity Hospital, we made a 3D printed nasal cavity replicate model, which is based on the CT scan of the patient that is representative for the condition in question. So I have here video material which is showing the assembly of the model. You can see that the model is composed of several parts, which makes the deposition study easier. When the model is assembled, you will see the outer nose, which is important to apply the drug. And you can see also at the back side the connector used for the connection of the model to the respiratory pump. We really believe that this approach will ensure obtaining more relevant data on deposition profile. Conclusively, describe the academic industry collaboration in the field of innovative nasal drug delivery systems and also supported by interdisciplinary teamwork has really the potential to encourage the development and commercialization of innovative nasal products, bringing benefits that could be translated into positive national growth rates. And this concept is in line with smart specialization strategy of Republic of Croatia. And in the end, I would like to present our joint publication, which I believe is actually just the beginning of this academia industry fruitful collaboration. And finally, I would like to thank Croatian Science Foundation for funding this project and all the participating investigators and collaborators on their immense contribution. And thank you all for your attention.
Thank you very much. We have one specific question related to this presentation. So I will ask this first before we move to the general discussion. So the question is in the chat, and it's what is the average decomposition time of the pectin gel, and what is the association of the decomposition with drug diffusion? And then the follow-up question, how did you decide to use pectin and not other polymers?
Okay, I will start with what I remember. So we actually made rheological studies, which may be found in our publication. We measured gelation time in a way that we actually mixed our system with the same volume of the simulated nasal fluid containing calcium ions. This ratio was selected due to the fact that actually nasal mucosa is covered by 100 microliters of the, let's say, nasal fluid, and the volume that was sprayed in the nasal cavity was pretty the same. So we mixed these two fluids in a volume ratio 1:1. And the concentration of calcium in this simulated nasal fluid was actually biorelevant concentration. And then we used the time gelling test, as I said, through our rheological assessment. And the other question was linked to the diffusion of the drug through the gel. So we used we used this in vitro release testing to see if the release of a drug will be actually slowed down due to the three-dimensional structure of the gel. And we found out that actually we use polymers like pectin, sodium hyaluronate, and gelangam. And the third one actually was the one that determined the rate of diffusion or the rate of drug release from the system. So we had a chance actually to, let's say, control release by optimizing the concentrations of the polymer in the system. I'm not sure if I answered all the questions.
Yes, and we see, so we have the message in the chat. Great, thanks. So that's it. Thank you so much.
Okay, but all the details may be found in the publication. So
okay, and we have another follow-up question. So I'm going to read this one as well. So this is, did you perhaps encounter an API which was highly incompatible for nasal drug delivery during your research? This is the first question. And then the second, is it possible for an active compound to have its stability decreased by being incorporated into a nasal drug delivery system?
Of course, both aspects are being studied through our studies. The first one, we do screening using cells, epithelial nasal cells, actually, let's say a model that can be used for simulating conditions in nasal mucosa. We used CALO3 cells, and first of all, we screen biocompatibility and then permeability of a drug through this, let's say, model barrier in order to screen the systemic bioavailability of drugs. Sometimes we wish that the drug is systemically available, and sometimes we just want localized effect. So that's the first question. And the second one was related to stability. The first system we made was a system of fluticasone propionate, and it is actually the drug with a low solubility. We tested the stability of the system over time and found no problem. But of course, this is one of the most important considerations in development of the system. So it is important to assess the stability, the need for buffering the systems, adjusting osmolarity, etc.
Thank you. And I think that, yes, so yeah, we have also the things in the chat. And actually with that, we can
I just
move.
want to thank attendance for the questions.
Thank you. And I think with this, we can move to the general discussion. And actually, the last question, maybe also to extend it to our industry speakers, because nasal delivery is something that's been, I would say, explored for a while. And I know that big pharma, has also been looking into it in some other aspects. And of course there are some questions that are inherent in the system, and then they have been actually posed in the chat. But also I would like to ask you about the studies, if they have been done about compliance, and about how is the nasal drug delivery being generally received by the patients.
Is this question for me?
This is question for you, but also for maybe for the industry partners. So for Maša, but also for Sandra. So anybody, yeah. Okay.
May I, is this regarding the part of the question if the active compound has its stability decreased by it being incorporated into nasal drug delivery system?
Yes. So this is the general applicability, I would say, of the nasal drug delivery.
I think here is more question about whether it's stable in aqueous solution, and you need to find, because nasal, if we are talking about solutions, we are working mostly with solutions, so I will address solutions. So a drug can be stable in a solution in a particulate pH range. You can change this by going into better pH range or adding some buffers or things like that. Or if it's sensitive to oxygen, adding antioxidants. So it's more regarding the metrics, its stability, than the route of application, by my experience.
Okay. And also, have you, has any research or studies been done on, I would say, compliance on the patient side? So when you are introducing a nasal delivery, how does it affect it?
Well, we didn't. We test if they understand the instructions. But will they use, and also we have studies that see if it can be used as intended to be used. But whether they will use it as often as they should, really we didn't make those studies. It's more question for health authorities if they are giving some product to a patient to check whether they are using it as it's intended.
Yeah. Okay. Thank you. Thank you so much. I don't see any questions in the chat. I do have some general questions to ask you, but we will reduce it a little bit because of the time pressures. And I would just really want to thank all the speakers once again for giving us all these different aspects of the innovation, of the manufacturing process, but also I would say about the different innovation that you can really bring by extending your product range with this value-added genetics, which was which was also mentioned as well. So the more we follow up and a question to ask everybody, it would be the same question that I asked Sasha first, and this is now to look back a little bit to this industry academia connection, and to look and really to look how how would you best optimize it and how would you utilize it. And we have seen an example, and actually we have seen an example of what is even goes beyond industry academia, because the hospital was involved as well. But there are also other situations, I would say, where this can be relevant, and so I would really ask our industry speakers, and I know that I think that Sandra is here still, and Maša. to comment, especially on this industry, on the industry academia angle. So, in which aspects have you found that this collaboration most effective, or what would what would be your recommendations for improvement in the field?
Okay.
Maybe Sandra first, if you are. I don't see everybody.
Hi.
Hi.
Yes.
Yes.
I'm here.
Yes. Okay, Sandra.
Yes, yes. For me, it was really interesting to see some of these examples of industry collaboration, especially this lecture from Anita. And I think it should be more and more of such examples, because we could end up with some really great products on the market with benefits to the patients. And I also think maybe when in industry we have, let's say, issue with some of the our products or some investigations, it would be great if we could also approach the university and maybe like outsource some part of our research in collaboration with university. It would be really helpful.
Thank you. And what was your experience in previous collaborations? Were they mostly about, I would say, formulation development, or did you bring people where you had some, like this investigating work what you were doing with the particles as well, so?
Yeah, mostly we were collaborating with universities for specific analytical techniques, which maybe we don't have in-house, specific instruments, or we do not have expertise in that area. And that was a great then collaboration to can approach the universities and quickly solve some problems.
Okay, thank you. And also then, the question, maybe the same question, but then extended to Jadran Galenski Laboratories and to Maša. So your take on industry academia collaboration. And I know that you are part of the collaboration that Professor Hafner was presenting, but I would also maybe comment wider on that.
Yes, we have more than that collaboration. That was just the beginning. Actually, each year the collaboration gets better and better. We in industry have learned to ask specific questions and to be more specific on what outcomes do we want from a collaboration. And also it's really important that we have a project manager or somebody that is looking for the being on time and within the budget. So that should be on our side if we ask for the collaboration, of course. But I'm really impressed. If you asked me this 5 years ago, I would be a little bit cynical, but now I'm very, very impressed about the collaboration, because apart from with Professor Hafner collaboration, we have more than one with faculty of pharmacy, but we also have very good collaboration with Faculty of Medicine in Rijeka, and we have a project, ERI project with them, and also with Faculty of Biotechnology. But as I said, collaboration is people, and people need to communicate openly and to be clear on what anybody can do in which timelines, and be open about it.
Thank you so much for these insights. And I would say it is a little bit of shame that we can't see reaction from the audience, since the videos are not on. What I will say that it really is a theme that you hear discussing in industry, and this is this emphasis on people, and really to all the young researchers, early career researchers in the audience, to understand that sometimes the timelines can be very long because you need to start maybe with a smaller project and then follow up with a larger one and really have to build your credibility towards any partner. It doesn't have to be an industry partner. And also, I would extend this question to Professor Hafner, because you mentioned the same, that you've involved, of course, the industry partner, but also the hospitals. So you are really now in a position to coordinate what is a very complex collaboration. And maybe from your perspective, what did you find the most important factors to be in place to actually be able to set it up, to apply, to win the project, but also now in the implementation phase?
Mm-hmm. Well, I think that the major point is in a high need for advancing and implementing of in vitro tools in a directed development of drug delivery products, drug delivery systems. So what we provided in our project is a systematic approach to in vitro characterization tools in order to screen therapeutic potential of the formulation candidates and to select the leading candidates that can be, of course, analyzed in subsequent phases, included maybe in vivo aspects of characterization. So I think that was the...
You would say good project design, right?
Well, I think that this is also the strength of the collaboration, because one of the possible results, one of the results of this collaboration might be the implementation of such approach in development of pharmaceutical formulations.
Mm-hmm. Okay, thank you so much. Please, I want to, again, members of the audience, feel free to ask questions. There is still a little bit of time to ask new questions. And I will move to maybe another aspect that I would like to tackle while we are all together. And because we heard about so much issues of the industry has, so this was both from the presentations from PLIVA, but also from JGL, that are not necessarily related to active ingredient, or that are not maybe related to the biochemistry of the active ingredient per se. In the first presentation, we heard really about the supplier discussions and the detective work and really going back to the supplier and having to resolve some issues. And then in the second presentation, we also heard about this cost of goods and services and manufacturing efficiency. That was also very much an important factor. So again, questions for the industry speakers and anybody can also feel free to jump in. Is to which extent are your R&D activities, but also then industry academia collaboration activities cover these other aspects? So not necessarily biochemical aspects, but also the improving the efficiency. Through different ways of organizing work, from introducing new technologies, from maybe to introducing new information technologies, or automated system, autonomous system. Have you had exposure to that, and what was your experience?
Yes, certainly we try to make continuous improvements in all the phases, and meaning in the manufacturing process, try to put some little steps, little changes to have a better process. And also, I think that in pharmaceutical industry, this automatization of processes and this process analytical technology, it is not yet still so widely used as maybe in some other types of industries, because there is maybe some reluctancy to do the changes, to approach regulatories, to improve the processes. So it's important when developing new products to implement this from the start into the manufacturing process.
So it really needs to be planned from the very beginning. So maybe this is also the opportunity for those producers who are more active in the generics, but there is always a cost, I would say. And the question, the same question, or the similar question to Maša and to JGL, any R&D activities or activities later on that really tackle this efficiency of the manufacturing process and not expanding the product range per se.
Sorry, I would ask you, Vlatka, I'm sorry to repeat the question because I have troubles with connections. I didn't hear everything.
No worries, no worries. So the question was because your presentation was really about you were talking about expanding the product range and really evaluating generics, but at one point you mentioned the cost of goods and services, and you mentioned this importance of manufacturing efficiency and manufacturing process. So my question is also to you: how much R&D are you doing in that field? Are you also doing this R&D with in connection with I would say academia, and what are maybe the technologies or disciplines that are involved, because they don't necessarily, they can be bioprocessing, but they also could be information technology, autonomous systems, and so on. What we heard from Sandra, that it's not really that easy to introduce new technologies into drug production because of all the regulatory issues. So and you can also then comment on that aspect as well.
Well, it's not easy about regulatory, but the first requirement for any optimization is that you really know how you are producing your product or how a product is produced, so you can then suggest some lowering of the production, because mostly the cost of goods, there are cost of API, cost of excipients, maybe you need to find a better supplier, or you need to be vertically integrated. That means that somebody is producing, that you are producing for yourself. And the other is maybe to not have 20 steps, but to have 10 steps. And it's possible to have collaboration, maybe if we have a case study or something like that to invite. We had a case study on the faculty of pharmacy, and we asked students about possible optimizations or adding new products to the portfolio. But this would be really a thing that you do knowing your process very, very well, and your suppliers and everything that makes the production cost. This is talking about my experience, of course. Maybe somebody finds, I don't know, some other, maybe biosimilars could be a way of lowering the cost, but a very different way of thinking, because you are going around patents and giving medicines that are not so costly, but not by a traditional way of lowering the production costs.
And yes, definitely. And I would say, what you said, that this is your view, but this is definitely not just your view, because we had a webinar that was in a completely different field. So it was like an energy production system, and a speaker that came, it said, they said very much the same thing. So really you need to understand what you are doing, and then, but very much in very detail. And then from there, you can then move to optimizing into maybe forming a question to involve the academic partner, and then working on that.
Yes, there is a very interesting approach. It's called open innovation, where you disclose what need you have, and you disclose it openly or semi-openly. There are forums and applications for disclosing. And then you say, okay, I have this issue. Has anybody any idea how to solve it? Xellia Pharma is developing products like that, and other companies also approach their problem. It's another way of thinking. When you are come to the limits of your, the problem cannot be solved on the level where it happened. So sometimes you need outside perspective. But it's a little bit, not a little bit, it's a new approach for a very conservative industry like pharma. But it will come.
It will come, and we do see more and more platforms that, like the one that you mentioned. There are also, I would say, like open calls by big pharma, but maybe in a different area, maybe looking at whether it's targets or but it does signal a little bit this openness to collaboration and to work together. So with that, I don't see any new questions in the chat. So I would really like to close this session to thank you all for taking part. All the speakers okay, we have a question. So there is a quick question which we will ask. And this is importance of IP protection. And this is a very good point, actually, because when we are doing in industry academia collaboration, IP comes in very often. There is, of course, the need of the industry, of the academia to publish, and there is, of course, the need of the industry to push drug development, product development, and to maintain confidentiality to some extent. So I would say a final question then for all the speakers, if you can comment on this specific issue on IP, and what is your approach to this question in collaborations?
May I, Vlatka?
Yes,
because
yes.
I think it's not mutually exclusive. It's just a thing about timing. As we have 2 or 3 ongoing collaborations, and we just agreed that we will first file a patent, and then academia can make as many publications as it wants. It's really in our interest. But to be honest, patents are a way of knowledge sharing. 20 years ago, you would go to Science Direct or other types to find new information and new knowledge. Today, you are going to patent databases and finding relevant information. And it's the same level. You need to check what's written in a scientific journal as well as in IP. But I would say that today the knowledge sharing has shifted to the patents.
Patent literature. Okay, thank you. So any Sandra or Professor Hafner, if you have any comments to the same question? If not, then I think we will leave it at that. So yes, IP is important. However, it is not a block or a stumbling stone to cooperation. And with the little management and communication, it can really serve the dual purpose and really push both the scientific publishing and as well ensure the necessary protections. And so with that, I would want to thank all the speakers once again for joining us, for really sharing these insights. it is very it was a very, I would say, open presentation and open discussion, and I'm sure it will be very useful for our audience, especially for early career researchers, to understand these insights from the industry side. And also special thanks to Professor Hafner for sharing the details of her project, and in both the scientific but also on how was it organized. I do thank all the participants, so all of you for your patience, especially in the beginning as we had these technical problems. We persevered, and I would say the end discussion and all the presentations I really overcame this glitches in the beginning. With that, I would really like to think once more, if you are a participant and if you are taking part in the winter schools, other webinars, or in the course, we will all see you again in different sessions, and there will be more chances to discuss. And with that, with one more thanks to all of you, I really do wish you all the best and all of in your work and in your life, and a pleasant evening today.
Thank you. Bye.
Thank you.
Bye-bye.
Bye-bye to everybody.
Thank you. Goodbye.